Hey guys, Miss Cote here, and welcome to another episode of the Heredity Unit. In the previous videos, we looked at how Gregor Mendel put forth two laws to explain how traits are passed down from parents to offspring through the combination of two alleles. We also saw some deviations from the regular dominant recessive inheritance. In this video, we're going to look at how Mendel's work was expanded on to incorporate the chromosomes that the genes are located on. So let's get started. Keep in mind that when Mendel did his work, the term gene did not exist. With the eventual discovery of chromosomes and scientists working out the processes of mitosis and meiosis, the chromosomal theory of inheritance came about. As you can see in this diagram, the Y and R alleles from the dihybrid cross we looked at in an earlier video are now physically placed on chromosomes, and we can actually track how these alleles separate and independently assort. I've said it before, while Mendel had no idea about genes and chromosomes, his work and theories were spot on and clearly ahead of his time. One scientist who still wasn't quite convinced about all this was Thomas Hunt Morgan. Morgan did his work with fruit flies, also known as Drosophila. Like Mendel's choice of pea plants, Drosophila were a good animal model for genetics because they produced a lot of offspring, they reproduced pretty fast, and they only have 4 pairs of chromosomes. Through his experiments, he provided the evidence showing that genes are located on chromosomes. As he did with many crosses, he came up with vocabulary and symbols for the flies. For example, the phenotype that was most commonly found in natural populations is referred to as the wild type. Keep in mind that just because something is the most abundant in a population doesn't necessarily make it the dominant phenotype. Any phenotype that appears that is not the wild type is called a mutant. For example, the first mutant phenotype Morgan discovered was white eye color. So let's look at Morgan's experiment. Morgan crossed a red-eyed female, which is the wild type, with a white-eyed male, which is a mutant. All of the F1 generation had red eyes. And when the F1 generation was crossed, the F2 generation showed a 3-to-1 ratio. Now that's what you would expect to see with Mendel's crosses. However, none of the white-eyed flies were female. Only the male flies had white eyes. That intrigued Morgan, and he eventually determined that this gene must be found on the X chromosome, and this supported the chromosomal theory of inheritance. So looking at the model that Morgan came up with, if this gene is on the X chromosome, females who are XX would have 2 alleles. Notice the symbols being used. The letter represents the mutant, so we're using W for white eyes. The wild type is designated with a plus sign next to the W. Keep in mind that sometimes you may see just the plus sign. That would designate it also as being wild type. So the female is true breeding wild type and will have red eyes. The male fly, who is XY, only has 1 allele because he only has 1 X chromosome. Notice there's nothing on the Y chromosome because the gene is not found on the Y, only on the X. And since he is a mutant, he has white eyes, he has a W with no plus sign. So when the Punnett square is completed for the F1 generation, the female flies are heterozygous for red eyes. They can also be called carriers, since they carry the allele for white eyes and can pass that allele onto their offspring. The male flies are all red-eyed because they have the red eye allele. Note that males can only be either red or white. There's no way they can be heterozygous because they only get 1 allele, and that 1 allele will determine their phenotype. And when these F1 flies are crossed, we see how we get the 3-to-1 ratio. The 2 female flies have red eyes, although one is homozygous and the other is a carrier. And 1 male gets the red allele while the other gets the white allele. Just be careful. Make sure you read what the question is asking. If they're asking how many flies are red, you would say 75%. If they ask how many females, then it would be 100%. If they ask how many males, it would be 50%. This is where you may need to distinguish between males and females. And not to go too much into it, but please know that for those animals that have a chromosomal basis of sex determination, because not all animals have this, It's not necessarily the X and Y system that humans and mammals have. There is also an XO system where females are XX and males are XO, a ZW system where females are ZW and males are ZZ, and a haplodiploid system where in bees, unfertilized eggs can develop into males and fertilized eggs develop into females. Pretty much all of the questions I've seen regarding sex-linked traits have focused on the XY system. I just wanted you to know that there are other systems, just in case. So if a gene is on a sex chromosome as opposed to an autosome, it's called a sex-linked gene. If it's on the Y chromosome, it's a Y-linked gene, and if it's on the X chromosome, it's called an X-linked gene. The reason why we see more questions involving X-linked genes is due to the size of the two chromosomes. The X chromosome is a lot bigger than the Y chromosome and therefore has more genes on it. Most of the genes on the Y chromosome only deal with sex determination. Classic human X-linked genes are a form of color blindness, hemophilia, and Duchenne's muscular dystrophy, which are all recessive, by the way. Here are 3 examples from your book. They're using color blindness as an example, represented by capital N for normal and lowercase for color blindness. Notice that these N's are written as superscripts on the X chromosome, since this is an X-linked trait. And there are no superscripts on the Y, since it doesn't carry the gene. In the first example, we're crossing a homozygous woman for normal vision with a colorblind man. This results in all the daughters having normal vision, but being carriers, and all sons having normal vision as well. The second example is crossing a woman with normal vision, but is heterozygous, a carrier, with a normal male. This results in all daughters having normal vision, but there's a 50% chance that they will be carriers. And with the sons, there is a 50% chance that they could have color blindness. The third example is crossing a woman with normal vision who is a carrier with a man who has color blindness. For the daughters, there's a 50% chance that a daughter could have color blindness, with the normal daughter being a carrier. And there's a 50% chance that a son will be colorblind. Remember, for a recessive X-linked trait to be expressed, a female needs both recessive alleles while males only need one. This makes these types of disorders more common in males because of only needing one recessive allele. The last thing I want to mention is that just because females have 2 X chromosomes, this doesn't mean that proteins from genes found on the X chromosome get made twice the amount of males, who only have 1 X chromosome. To make things equitable, in females, one of the X chromosomes gets inactivated in every cell. This inactivated X chromosome is called a bar body. What makes this interesting is when a female is heterozygous for an X-linked trait, because depending on which X chromosome becomes inactivated, some cells may express one allele while the other cells express the other allele. One example that comes up often is in the tortoiseshell cat. For a female that is heterozygous, some patches of fur are orange and some patches are black, depending on which X chromosome is active. Male cats, since they only have one allele for the tortoiseshell gene, will only be black or orange, unless there is some kind of chromosomal abnormality going on. So for tackling sex-linked problems, you approach it just like any other problem, but there are 2 steps to think about. Not only do you look at how the alleles are being inherited, but you also need to consider the sex of the individuals. So figure out the genotypes of the parents. You need to write out XX for mom and XY for dad, and then place the allele you are tracking as a superscript on the X chromosome. When you set up the Punnett square, separate the sex chromosomes, and don't forget the alleles, across the top and along the side. When you fill in the boxes, remember to put in the sex chromosomes and the alleles they are associated with. And then look at the question that's being asked. Are they asking for general phenotypes, or are they asking you for a specific sex? That could affect the percentage of the answer. And that's where I'm going to end this episode. This takes care of the first part of Chapter 15. The next video will tackle the concept of linked genes, which is something you didn't learn about in honors biology. So even though I'll go over it in class, This is something you should probably pay attention to because this is a concept that College Board has seemed to like in the past, and I'm not sure what's going to happen on this year's exam. And as always, until the next time I see you, be a good human.